Welcome to our lesson on antibiotics, painkillers and drug development. The key questions for today are 1. What are antibiotics? 2. Which diseases can antibiotics help to cure? 3. What are painkillers? 4. Where do new drugs come from? 5. How are new drugs developed and tested? The key words you'll need to know are antibiotics, antivirals, painkillers, toxicity, efficacy. Antibiotics are medicines that can be used to treat bacterial infections. These include infections of different organs in the body and diseases such as pneumonia. A common antibiotic is penicillin. Antibiotics destroy bacteria or inhibit their growth. They were originally made from a type of fungus and can be administered in the forms of pills, creams, and injections. Antibiotics are specific to a certain type of bacteria. This means that for a certain infection, a specific antibiotic is required. Antibiotics can only be used to treat bacterial infections. They are ineffective against diseases caused by other pathogens. For example, they will not work to treat flu. This is because the flu is caused by a virus. Antibiotics have been used since the 20th century and have greatly reduced the number of deaths caused by bacterial infections. However, some bacteria has become resistant to antibiotics. This could be caused by overuse of broad-spectrum antibiotics. These are antibiotics that are non-specific. MRSA is a bacteria that has become resistant to antibiotics. This is a worry, as if more bacteria become resistant to antibiotics, we will not be able to treat more and more infections, leading to an increase in the number of deaths from bacterial infections. Antibiotics are only prescribed if necessary, and specific antibiotics are used for different diseases. Patients must take the full course of any prescribed antibiotics to ensure all bacteria are killed. This will help to prevent bacteria becoming resistant to the antibiotic. Scientists are also trying to discover new antibiotics. Task: what type of infection can antibiotics be used to treat? Antibiotics can be used to treat bacterial infections. Extension: why has the development of antibiotics not reduced all disease-related deaths? Antibiotics are only effective against bacterial infections. They will not work against diseases caused by other pathogens or non-communicable diseases such as cancer or coronary heart disease. Other types of medicinal drugs include antiviral drugs, which treat diseases caused by viruses. Viral diseases are difficult to treat with drugs. As the virus invades body cells, this means that the drug must also have to attack the body cells, so can cause damage inside our bodies. Painkillers do not kill pathogens or treat any cause of the disease. They just relieve pain and fever. These would be symptoms, but not causes of the disease. Task: draw a table to outline what antibiotics, painkillers, and antivirals do. New drugs are regularly discovered. They often come from plants or microorganisms, but can be made in a laboratory. This is one of the reasons that it's important to maintain biodiversity, as plant and animal species could hold the key to new antibiotics. Here are some examples of drugs that have come from natural sources. Aspirin, a common painkiller, comes from willow bark. Digitalis, a heart drug, comes from foxglove plants. Penicillin, an antibiotic, comes from penicillium mold, which is a type of fungus. Existing drugs and new compounds, discovered or made in labs, are used to research new medicines. Chemists working in the pharmaceutical industry test and adapt the compounds to make more effective drugs. Task: draw a line from each drug to show where it originally came from. When new drugs are discovered, they go through a rigorous process of testing before they are able to be given to patients. Extension: why do new drugs need to be tested? What are they tested for? New drugs must be tested to ensure that they are safe, effective, and do not cause bad side effects. They must also be tested to determine safe dosages. Task: make a flow diagram to detail the steps in drug testing and development. The first stage in testing a new drug is preclinical testing. This involves the testing of a drug on individual cells and tissues, as well as computer simulations. During this stage, a drug may be tested on animals. The main things established during preclinical trials are whether the drug is toxic, so if it is poisonous or not. Obviously, if it's poisonous, it will not progress to stage 2 of testing. The efficacy of the drug. This means how well it works. If it doesn't work, it will not be tested further. The dosage. How much of the drug is needed to have the desired effect. Stage 2 in drug testing is clinical trials. This is when small doses of the drug are given to healthy human volunteers. This is to confirm the safety of the drug. Stage 3 is a second clinical trial in which healthy volunteers and volunteer patients are given different amounts of the drug to determine the optimum dosage. Stage 4 is wider clinical trials, where the drug is given to a wide range of patients and the efficacy is monitored to see how effective it is. Comparisons are made between the new drug and existing drugs, as well as placebos. Placebos are tablets that contain no medicine. Sometimes the psychological effect of taking a medicine makes people report feeling better. The use of a placebo allows the efficacy to be tested without this bias. Sometimes in these trials, the doctor and the patient are not told which drug they are taking. Only the scientists conducting the study would know. This is called a double-blind trial and eliminates any bias from the doctor or the patient. Any findings from drug trials are peer-reviewed by other scientists and are published in journals. Independent research is better than research done by the drug manufacturers, as they may be biased and try to make results look more favourable. Task: state 3 reasons drugs need to be tested. Drugs are tested to ensure they are safe and not toxic, that they work, and to see what dosage is required. Extension: how can bias be avoided in drug testing? To avoid bias, drugs should be tested by independent companies, not the manufacturers. Placebos and double-blind trials should be included.